Professor Karl Herrup, thank you so much for joining me today.
My pleasure.
We're going to start with a rapid fire round, and we are just discussing the fact that it's impossible for scientists to give brief responses to questions. But we'll try and do what's never been done. And so, to kick us off, globally, roughly how many people have dementia?
Globally, it's over 50 million was the last figure I saw. It waxes and wanes. It doesn't wane so much, mostly waxes.
Do more women or men have dementia?
More women than men absolutely have dementia. Part of that is because they live longer, but part of it is for reasons directly related to the biology of gender, and we don't fully understand that.
What proportion of dementia cases get labeled as being Alzheimer's disease?
Figures that get tossed about are 70% plus minus.
And what proportion of the risk of somebody developing dementia is thought to be explained by genes?
Oh, that's the number that's thrown around is again around 70%. I'm not a subscriber to that view, but that's a personal feeling.
I think we might be on the same page there. What proportion of dementia cases are thought to be preventable then?
Preventable?
Mhm.
Uh, I don't know. I'm going to take a pass on that question. I guess it depends on when you start. Um,
Yeah. Just to throw out a number, I think the Lancet Commission last year suggested 45%. But again, trying to come up with specific numbers, I think, is a very difficult thing to do.
right.
Alzheimer himself and his mentor, Emil Kraepelin, thought that brain morphology drives brain function. Do you believe that form fundamentally dictates function?
No, I don't. I do believe that form has an impact on function, but I don't think it dictates it.
What are the amyloid protein aggregates that accrue in Alzheimer's?
What of them?
What are they? Sorry.
What are they? They're uh, a chemical goo that forms because of structures called beta pleated sheets that causes roughly 40 amino acid peptides to kind of stick together and make a clump.
And is amyloid present in the brain of all people listening to this episode right now?
Every single one of them.
Should they be worried?
No.
What are the tau neurofibrillary tangles that accumulate in Alzheimer's as well?
They are twisted filaments of a hyperphosphorylated normal cellular protein called tau, gene known as MAPT.
The amyloid cascade hypothesis posits that brain amyloid initiates a kind of domino effect in which neurodegeneration propagates through the brain. In a word, do you think this is correct?
That word would be no.
Is your impression that the proportion of Alzheimer's disease researchers that believe this is increasing, decreasing, or staying much the same?
That's actually hard to know. I think skepticism is increasing. I think if you put a gun to somebody's head and said believe or no believe, the proportion might be staying fairly constant.
Your book is entitled How Not to Study a Disease. Was the net reaction of your Alzheimer's disease research colleagues positive, negative, or neutral?
Oh, well, actually, let's start with the response of the young people who I speak to. Most of them kind of take me aside and say, thank you, I thought I was crazy. And as for my Alzheimer colleagues, they've adopted the strategy of ignoring me to death.
Oh dear. Are anti-amyloid monoclonal antibodies proposed to treat Alzheimer's safe and effective?
Are they? No, they're neither safe nor effective.
Which class of medications do you think has the greatest potential to treat Alzheimer's?
We have had for several decades now cholinesterase inhibitors. One of the brand names is called Aricept. They are in people who can tolerate the drug, they're reasonably effective at relieving symptoms. Other than that, We have memantine, which is a weak drug, but nonetheless has modest effects. And I would say that's about it.
Is there a lifestyle domain or habit that you think has an underappreciated role in preventing Alzheimer's?
Underappreciated, I'm not sure. We're pretty clear that aggressive control of blood pressure is very effective in staving off the onset of dementia. Diet can be altered in a way that statistically staves off the onset. Specifics there are hard to come by. Exercise, which probably relates to heart health and blood pressure control, also seem to be effective. So yes, lifestyle choices can make a difference.
I'm going to ask the same question about environmental exposures. Is there an underappreciated exposure that you think either increases or reduces risk of Alzheimer's?
I think that's harder to pin down. There's data suggesting it's true. The data set itself is not as extensive. So underappreciated, maybe understudied, yes.
I'm going to preempt this question by saying that I myself am a contrarian in some ways. On your Twitter, you describe yourself as a professional iconoclast. Would you describe yourself as a contrarian, given that your book runs counter to the popular narrative?
Yes, I would call myself a polite contrarian. And the politeness at least gets me entrance into some of the more august meetings that I might otherwise be excluded from.
Has the field of research meaningfully progressed since your book was first published, which is probably coming up to 5 years ago now?
I'd say yes. I mean, good science gets done. Whether I would have done those particular experiments is a personal choice, but rock solid science is continuing to be put out under the heading of Alzheimer's.
And final question. Is the state of science in the US right now the most troubling that it's been in your career?
Oh, yes.
I thought that might be the case. I worried that might be the case.
Don't worry, it's an absolute fact.
Well, I'm very impressed by how punchy your answers were. I
I
was
was going to
expecting
say,
to be here for the next 10
I had
minutes.
a second cup of coffee, so I don't know. Let's roll.
So feel free to elaborate more on your answers from now on. You've put forward that an inability to coherently define Alzheimer's has hamstrung the field from making greater progress. Why is that so? And how would you personally define Alzheimer's?
Oh, I struggled with this question in the book, and I continue to struggle with it. And I'm still not sure I have even myself reached a satisfactory answer. I tend to be a big partisan of defining the disease by its clinical symptoms. That view is contested, and I would argue reasonably so, by those who say, well, every disease has to have some sort of biological underpinnings. So if we can define that biology, we should use that biology as a more definitive way of defining the disease. My personal feeling is that's fine, but that requires that we have a solid understanding, a solid agreement on that underlying biology. And basically, I spent a whole book saying we actually don't have an agreement on that underlying biology. Therefore, at the very least, it's premature to use markers such as amyloid, tau, and what have you as definitive markers of what we call Alzheimer's disease. Other than that, I think a huge problem, and this is generally acknowledged in the field, even by, rabid amyloid partisans, virtually every case of dementia comes with what the field calls comorbidities, which means that there are signs of other disease processes in addition to the deposition of amyloid and tau. So rare is the case of an Alzheimer brain that comes to autopsy with only signs of amyloid and tau and no other deficits.
So returning to your personal definition, what are some of the clinical features that you would witness?
Partly it's disease progression, so it would be age of onset after 65. So we're going to put aside for now the aggressive genetic forms of the disease and speak mostly of the far more common, what's known as sporadic form. So age of onset after 65, progressive loss of short-term memory, evolving of behavioral symptoms such as depression, apathy, and then progression through more and more serious dementia. And really, at the end of the disease, persons who suffer with it are literally immobile and need to be bedridden at the last stages. So, and I guess I would include, because with modern imaging methods, this could be determined, that there is brain shrinkage that would be relatively uniform across the neocortical areas.
Do we see the same type of brain atrophy across different neurodegenerative conditions,
Sure,
such as
we do.
Parkinson's?
Parkinson's, no, we don't. But there are certainly other conditions where the brain will shrink. Even the brain of an alcoholic might be seen to shrink dramatically by old age.
I was going to say, we're talking about brain atrophy in a condition which increases exponentially with age. Obviously, there is some brain atrophy that occurs during aging, but what you're referring to is accelerated brain atrophy relative to peers without the disease.
I actually don't know the specific numbers, but I think brain atrophy measurable at a gross level, such as imaging with MRI. I don't think that is a prominent part of aging brain, which is why I subscribe to the idea that Alzheimer's is a disease process rather than just an accelerated aging phenomenon.
Okay, that's interesting. We'll return to that later for sure. You mentioned familial Alzheimer's there and
Mm.
distinguishing it from the sporadic form of the disease. In your book, you suggest that we could reclassify those two as type 1 and type 2 Alzheimer's, much the
Yeah.
way that diabetes has been classified. What's the rationale behind that?
Like type 1 and type 2, the clinical symptoms are similar, the organ systems affected are similar, but we know for a fact that the underlying biology has the underlying pathology has a different origin. So, going to the case of Alzheimer's, we know that these familial forms have not only an earlier age of onset, almost by definition, they have to have a different biological origin because they originate from a very specific change in a very specific region of our DNA double helix. So, by definition, they're different diseases, and that applies to diabetes as well. Type 1 diabetes, there's a genetic change in the insulin receptor pathway components, and that would be the equivalent. Sporadic Alzheimer's would be the equivalent of type 2 diabetes, which is heavily affected by dietary choices during your lifespan and other environmental factors. So I see the analogy as pretty strong.
We might come back to some of those gene mutations that
Mm.
contribute to the familial form later.
Just to kind of give a shout out to my current work. We've been doing work recently in experimental systems that really underscores the fact that from a neural network point of view, understanding how neurons talk to each other, familial and sporadic forms of the disease are fundamentally different in how they impact the underlying neurobiology.
Do we see that at the level of brain structure as well? When you mentioned atrophy earlier, you spoke about the atrophy being global, so potentially affecting all the different parts of the brain.
Yep.
In familial Alzheimer's, is that the case too, or do we see selective atrophy of certain regions?
It's a good question, and I'm going to punt because I don't actually know what the brain volume does in genetic forms of Alzheimer's. I know the pattern of amyloid deposition is not identical to that found in the sporadic cases, but I'm going to take a pass on the volume. I should know that, and I don't.
Just to put this into context, about what proportion of Alzheimer's cases are familial? It's a small proportion, isn't it?
It's a very small proportion. It's certainly under 5%, and possibly as low as 1%.
There are different types of dementias. You've got vascular dementia, frontotemporal, Alzheimer's, mixed, etc. And there are two that come under the Alzheimer's umbrella. Do you think that might have actually done us a disservice? And if from the start we had treated them as discrete diseases, we might have gone about things quite differently.
I think that might have helped. I think the real problem is the sporadic forms and the effort to equate them with the genetic forms, so that studying a mouse model in which a gene has been engineered to mimic the human genetic forms of Alzheimer's and call that a model that will allow us to understand sporadic Alzheimer's. There, I think we may have been led astray.
And so, given the heterogeneity of these different types of dementia and neurodegenerative disease in general, if you're approaching categorizing these diseases with a blank slate, we didn't have our existing designations in place, how would you have approached that?
Good question. I would approach it, again, as I said before, I would approach it from the clinical symptoms. I think as a basic researcher, I'm much more comfortable considering that all these diseases are multifactorial in their final symptom presentation. And so drawing, clear, sharp boundaries between them is only moderately informative and may not be really helpful in terms of getting at the root causes of the dementia itself. Parkinson's, for example, comes with at the end stages of the disease, very often comes with a form of dementia. Understanding that dementia is different than understanding the tremor and the motor signs that come with Parkinson's. They undoubtedly come from a different biology. But you're beginning to see the complexity because I'm trying to tease out what causes the dementia in a condition like Alzheimer's from what causes some of the other symptoms that are maybe unique to the disease. So I think dementia is really the sticking point here. Maybe we need to just pull that out as a separate area of study, and make that our cause.
Were we to envision this, we might see it a bit like a Venn diagram, where there's a lot of overlap between these different neurodegenerative conditions.
Yeah, correct. And even more scary, I've been describing, dementia as almost an emergent property of the aging brain. And that's a scary concept because it means that no one component of the aging brain is either predictive or causative. It's the entire ensemble of events that happen during brain aging that allow the symptoms of dementia to reveal themselves. That doesn't fit anybody's pharmacological model, and so it makes life difficult.
Does chronological age predict Alzheimer's more strongly than it predicts other neurodegenerative conditions?
No, I don't think so. It is a strong predictor of vulnerability, but I think with age, your brain becomes more and more vulnerable of tipping into any of a number of different pathologies whose biology no doubt differs, but many of whose symptoms will come out looking very similar.
Aging might be at the core of your answer to the next question. Towards the end of your book, you describe what was then your current conceptualization of Alzheimer's and how we can think about the progression of the disease based on a series of analogies that I found compelling. How would you explain the biology to a smart 12-year-old who's interested, just so that before we get deeper into the details, we
Right.
can put it into relatively easy to understand terms? And has your conception changed much since the book?
The conception hasn't changed much. Looking back on that model that I tried to put together at the end of the book, I still think I would still stick with the idea that the activities that the brain goes through during early life in many ways presage what the vulnerabilities of that same brain are going to be as it ages. And that was the idea of what I called burn-in. That was frustrating because by the time I wrote the book, CRT screens, which is where burn-in actually happens, were almost eliminated in favor of LEDs. I wanted to go out and take a picture of a screen that had burn-in, and I literally could not find one. But anyways, to get back to the more serious point, if I've changed at all during in my sort of perspective on this disease since the book was written, it's to appreciate even more, which may strike you as odd given the emphasis I put on it, strike me even more that aging really is what we need to be studying. And so, in fact, and I swore I would never do this, I've started to put together a second book.
Ooh, it's like
And
self-harm.
yeah, huh? Well, we'll see. Right now it's still in the larval form. The whole premise of the second book is going to be rooted in aging.
Great. Well, I'm looking forward to reading that.
Yes, I'm looking forward to being able to read it too. Writing is just, it's a labor of love. I mean, you've got to be completely devoted because the pain is extraordinary.
Aging is a kind of unifying theme that runs through all the different processes that contribute to Alzheimer's, in particular, perhaps the sporadic form of Alzheimer's. But how does that tie into that conception that I mentioned that you detail in the book? Could you just highlight some of the key ideas in what you propose in your model of Alzheimer's?
To go back to the CRT analogy, when you have a new old monitor and you turn it on for the first time, it's bright and shiny and all the colors are true. But the more you use it, the more the individual elements in that screen begin to lose their integrity. And sometimes they can blank out totally, so you have a blank spot. But the analogy I tried to draw in the book, and I'm still thinking this does apply, is that a particular pattern of use in very, very small but incremental amounts remains embedded in the actual function of the screen. And that's what I call burn-in. So the old CRTs, if the classic would have been in an airport, where you can see where all the times were, even if you can't read the individual numbers, because at that point in the screen, for years, the monitor had tried to express a couple of numbers to tell you what time your plane was leaving. Well, it leaves an indelible mark ultimately in the screen, even when it's off. And that's my analogy, that it's the pattern of use that sets up what, I mean, and here the analogy gets a little weak, but would be a vulnerability, a structural failure to accurately portray color or intensity. And that's the analogy to our brains, losing the ability to accurately function, either in terms of executive processing, like doing a complex task, or memory, recalling a specific short-term event.
When you refer to use, I don't think you're just referring to participating in tasks that require your brain to contribute, whether that's working on a work project or playing the piano or walking. I think you're also referring to cleaning up damage as a result of various different exposures. Everything that your brain has to do. And so the type of burning that you're speaking about, it can affect many different structures in the brain, not just
Correct.
neurons, not just glial cells, it's the vasculature, it's everything.
It's everything.
And I suppose that some of this also ties into the loss of neuroplasticity, or the brain's ability to remodel itself. Getting into some of the details now, you mentioned amyloid and tau already, and those are proteins that get folded in certain ways and then can clump together into aggregates. And this brings up proteostasis in general. And actually, if you look at the so-called hallmarks of aging, then loss of proteostasis is one of the ones that have been put forward as being a driver of aging.
Yes.
So I suppose we should probably explain what proteostasis is and why it matters.
So proteostasis is a sort of a broad umbrella term that talks about maintaining the, let's call it the structural integrity of your individual cellular proteins. What we typically focus when we talk about proteostasis, we talk typically about the process of degradation. More specifically, what that refers to is that proteins are like any other piece of biology. They get old, and they break down. And when they break down, the cells have come to recognize that it's worse having a malfunctioning protein than no protein. And so there's an elaborate system within our cells that lets the damaged proteins be targeted and sent off to various compartments in the cell where they're broken down into their constituent amino acids, and those amino acids are reused to build new fresh proteins. So that whole process of recognizing defective proteins, targeting them for degradation, breaking them down into their individual parts, and resynthesizing fresh and active proteins would be at least my definition of what proteostasis is.
And importantly, it's the structure of proteins that affects their function.
Correct.
And that's relevant to various different factors that contribute to Alzheimer's. One example that comes to mind is variation in the apolipoprotein E, APOE gene.
Correct.
That influences the structure of that, and then downstream of that, many different processes in cells. But if we focus on the amyloid cascade hypothesis, can you just outline what the hypothesis is and some of the key actors involved, including amyloid precursor protein, the enzymes that cleave it, and so on?
Right. It's known many variations over the years, but it always falls back on a relatively linear pathway to describe the well, let's call it the pathogenesis of Alzheimer's disease. The core of the hypothesis is that the disease process starts with abnormal accumulation of a small piece of a larger protein known as the amyloid precursor protein. We'll come back to that, but those fragments then, with age or with altered genetics, accumulate in numbers that the proteostasis system, which we just talked about, it becomes overwhelmed. They have the property of being somewhat sticky. And so if they're not destroyed, they tend to form clumps, and the clumps grow. And as the clumps grow, they start to be recognized by the innate immune system of the brain, interprets them as a foreign invader, begins to mount an inflammatory response. In addition, the hypothesis holds that through a mechanism that I think there's not complete agreement on, this then triggers the abnormalities in the tau protein and its phosphorylation. So secondarily to the clumping of the fragment of the amyloid precursor protein known as A beta or amyloid. Secondary to that, clumps of this phosphorylated tau protein start to form, and it's all downhill from there. There's neuronal dysfunction, neuronal death, and ultimately that is the cause of the dementia. So you have a linear pathway from too much amyloid, inflammation, tau abnormalities, neuronal dysfunction, and death. And as I said, over the years, different aspects of that pathway have been stressed or de-stressed, but that's the essence of the hypothesis.
You mentioned amyloid precursor protein there. I
Mm.
understand that much of your lab's work centers on that protein. You led some very interesting work on its roles in regulation of axon morphology.
Mm-hmm.
You might explain what axons are. That was published in the Journal of Neuroscience a couple of years ago. What did you find? And what do you think the precursor protein's functions actually are?
So that's a really good question. And one of my indictments of the field is that for all that we focus on this little breakdown peptide, A beta amyloid, we've largely ignored, not completely, but largely ignored what the normal function of this large membrane protein is. I like to throw up a slide when I'm giving a talk. It shows a little ball and stick diagram of what the protein looks like. And I flag the portion of that protein that is actually the amyloid peptide itself. And
Tiny.
it's about 5% of the total protein. So it kind of strains credulity that this whole protein is there to do nothing except create amyloids. It's got to be doing something. And in fact, we know that it is. So it has a huge role in the development of the brain. It impacts neuronal migration. It impacts synaptogenesis, the formation of connections between neurons. So it has a huge role in development. And what our work showed was that it also has what we could call a homeostatic role in normal neuronal function. So when a neuron gets a little bit more active than normal, that activity triggers an increase in the levels of the amyloid precursor protein. And that increase causes the structure of the axon to change in such a way that it becomes less easy to create an electrical signal.
Mm.
And that becomes almost like a governor. So if you get too much activity, APP goes up. APP is the abbreviation for amyloid precursor protein. APP goes up, it changes the axon structure, and that quiets the neuron back down. So it's a protective mechanism.
It's like a thermostat.
Like a thermostat.
That's very interesting. I think a lot of people have been interested in APP, amyloid precursor protein, in part because mutations in it are genetic drivers of the familial form of the disease, along with mutations in a couple of other genes, presenilin 1 and 2, which influence the activity of one of the enzymes, gamma secretase, that cuts APP. This brings to mind some of your comments earlier, because it seems to me that APP and the presenilins, and perhaps amyloid in general, and the mouse models that have been developed to mimic amyloidosis are most relevant to the familial form of the disease. Do you think that's a fair comment?
Oh, I think it's totally fair. And as I was saying, the work that we're just now submitting for publication at a cellular level argues that they have very different impacts on the function and even the structure of neuronal networks. So yes, I think they're very different conditions. And I think it plays into what I said before, which is that these genetic forms of Alzheimer's, whether the mutation is in the APP gene or in the presenilin gene, they're going to impact the brain from conception. So the entire developmental process of the brain, which is very, very formative in its ultimate ability to function, will have been undertaken in the context of having a mutant APP gene. Now, our biologies are very clever, and they can almost always work around a problem like that. But the workaround leaves the brain vulnerable in other ways that may not be apparent right away, but will be apparent as the brain ages.
Regarding amyloid beta, these peptides that APP gets cut into, people have conflicting opinions about whether all amyloid is bad. And some people who are willing to question convention have put forward that amyloid likely has various roles in normal physiology. As we mentioned, it's present in everybody's brain, and people have pointed to roles in immune function and various other processes. Do you think that it has some roles in a healthy brain? What do you think those roles might be? And also, do you think we need to distinguish different amyloid peptides? Are they all made equal? Are shorter peptides different from longer ones?
Let me start with the overarching question, which is there a normal function for this little amyloid peptide? And I think the answer is probably yes. I have a friend and colleague, Alberto Espe, who is a very strong partisan of the idea that, in fact, the problem in Alzheimer's is too little of the A beta, the amyloid peptide that is one of the longer forms. It's A beta 42, because the 42 stands for the number of amino acids in that peptide. And the evidence for it is somewhat compelling, including some of the clinical trials that were done for the monoclonal antibodies. So does A beta have a normal physiological function? We knew it had a normal physiological function. It is absolutely a synaptic modulator. How that folds into the normal function of the brain, I don't think we're entirely clear. Does it have a role in Alzheimer's? If Espe is right, then it actually does, because there's not enough of it, which turns all of our Alzheimer's therapeutic approaches on their head. But, data's data. And then now, of course, I'm forgetting what the second part of your question was. So let's move on.
The second part of the question was
I'm allowed to
about
play the old card occasionally, so I'll play it for the first time here.
Well, I recognize the importance of cognitive stimulation to mitigating Alzheimer's. So my goal is to maximize the cognitive load of this conversation.
And I
Everyone
appreciate
listening too.
Yeah, thank you very much.
Yeah, the second part of the question was just about the different peptides and whether they differ in their effects. But I think you somewhat alluded to that anyway.
I did touch on that. Yeah, they do. It does matter.
In your book, you at length describe how the amyloid cascade hypothesis has failed some critical tests. Can you briefly describe what these tests are? I think you highlight three of them.
The two critical ones are this. If the hypothesis is true, then if you add amyloid to the brain of a healthy person, you should create Alzheimer's disease. That would be a prediction of the hypothesis. That is fundamentally not true. So 30% of perfectly cognitively intact individuals, people walking around, living their best normal life, if they're over the age of 65, they have a 30% chance of having amyloid burdens that would be consistent with the diagnosis of at least early stages of Alzheimer's disease. Do they have an increased risk of developing Alzheimer's? Yes, they do, but the increased risk is not dramatic. And in any event, those individuals can live for another 5 years. Only half of them will progress into some form of dementing illness. So at the very least, the presence of amyloid, it doesn't strike one as a biological mechanism. A risk factor, perhaps, but to say that amyloid deposits can wait 5 years before having a 50% chance of triggering a dementia, to me, that's a failure of the hypothesis. And then the mouse data suggests the same, which is that we can take mice and engineer them so they produce boatloads of amyloid and fill their brains with it. And they do end up with memory problems, but they don't end up with anything resembling the clinical symptoms of Alzheimer's disease. And as I point out in the book, more importantly, if you do things to get rid of that amyloid, the mice get better. They get 100% better. And that does not describe the human condition. So if we get rid of amyloid in the human brain, people don't get better. And in fact, the recent monoclonal clinical trials suggest they continue to get worse. So that's trial number one. Trial number two is if we take amyloid out of the brain, we should prevent Alzheimer's. Neurodegenerative diseases are nasty because you can't reverse damage once it's done, but you should be able to stop the disease if the amyloid cascade hypothesis is correct, because further damage should be blocked. And in point of fact, if you look over the years at the clinical trials that have been done, we've been able to reduce or virtually eliminate amyloid from the brain using various strategies. And in most cases, actually in all cases, people continue to get worse. In a tiny fraction of the cases, less than 5%, with very significant amyloid reduction, we can slightly slow the rate of decline, but people don't get better. So that's a failure of the second test. The third test would be that you should be able to block the formation of the peptide and stop the disease, and that doesn't work either. It's always hard to be polite at this point because what I want to say is, so why are we bothering with this hypothesis? But the polite way to say that is, okay, there may be some biological contribution to the total picture from amyloid, but we're going to have to put it in its place, and we're going to have to remove it. And here is the key part of my argument. We're going to have to remove it from the biological definition of the disease, and we're certainly going to have to remove it from the definition of the disease, which is where it stands right now.
To put something into perspective that you mentioned earlier, I think the increased risk that's associated with the high amyloid burden at baseline is roughly equivalent to carrying one copy of the APOE4 allele. Is that correct?
That is correct. Yeah.
Roughly a threefold increased risk, something like that.
Yeah, the numbers may vary. I haven't gone back into the weeds since I wrote the book. Maybe the numbers have changed, but that's roughly correct, that the risk is less than being an APOE4 homozygote.
And is it fair to say that every patient that has severe Alzheimer's disease will have a lot of amyloid in their brain?
Well, that, of course, gets all knotted up in the definition, because by definition, if they have a clinical picture, a set of clinical symptoms that looks exactly like Alzheimer's disease, but they don't have amyloid deposits in their brain, by definition, that was not Alzheimer's disease.
So if we rewrite definition to exclude the amyloid, then would that be true?
Well, then I would say we can go to the clinical literature and find out that when a neurologist diagnoses Alzheimer's, the pathologist gets to look or the radiologist gets to look at the presence or absence of amyloid. And by its absence, the neurologist was wrong 15% of the time. But I take that as evidence that the neurologist was right. And in fact, 15% of Alzheimer's dementia does not have amyloid deposits.
The reason I asked that is because I was thinking about tau tangles and the fact that they seem, but maybe this is another product of the way that Alzheimer's is defined, to be less specific to Alzheimer's compared with amyloid. So if we look across different neurodegenerative condition, then we see tau tangles popping up in various other disorders too.
Yes.
Is that a fair statement?
That is a fair statement, yes.
What do you think are some of the potential effects of the tau tangles in Alzheimer's pathophysiology, if any?
Good question. I find the tau tangles to be much more biologically entwined with the disease process than the amyloid deposits. So I said before, we can fill a mouse's head with amyloid and they have a few short-term memory problems, but no neurodegeneration. That's not true for tau. If we actually create genetic models that incorporate some of the mutations of tau that lead to neurodegeneration, we get neurodegeneration, and the symptoms begin to look much more similar. Not perfect, but they look much more similar than do their amyloid cousins. So tau contributes, but as you said, and I think it's true, those deposits are not disease specific. They're found in other conditions.
What do mutations in the gene that encodes tau cause? Which neurodegenerative diseases do they contribute to most?
So it depends on the mutation, of course, but they're linked to a form of dementia known as frontotemporal dementia. And more recently, and I think in a very exciting scientific advance, they're also linked in almost a continuum with amyotrophic lateral sclerosis, or ALS, or Lou Gehrig's disease. So very involved in neurodegenerative illnesses, which both by symptoms and by their underlying biology are different from what goes on in Alzheimer's disease.
Interesting. We spoke a little bit about brain atrophy earlier, and that is somewhat related to the loss of the volume and or numbers of certain cells in the brain. Those can include neurons. How much do you think that brain volume loss contributes to Alzheimer's? I know that it's also downstream of various other issues, but do you think that fundamentally that's a really important driver of Alzheimer's?
Oh boy, a very good question. And let me be a scientist for a moment and say the answer is yes and no. Yes, it's much harder to have a normal functioning brain if you take away a third of it. And yet, our brains are very, very plastic and are. Let me put it this way. I think the loss of a quarter of the brain volume, as we see in Alzheimer's, I think in and of itself, the brain could deal with that the network properties of the brain are robust enough that you may have some loose ends hanging out, but that the overall function of the brain would be just fine. So when I say yes or no, what I mean by that is that having less gray matter is going to strain the network to maintain function, but does not by itself cause the disease.
Do we know much about how the trajectory of network changes relates to change in brain structure?
No, we don't. In other words, it's a huge chicken and egg problem. So does the lack of function lead to the death of neurons, or does the death of neurons lead to lack of function? I don't think there's a clear answer to that.
Your lab's focused quite a lot on the roles of unscheduled cell cycle events in the loss of neurons. Can you summarize what the cell cycle is, and then what you found regarding how that relates to the viability of neurons?
It's a problem which I have to say I have not worked on recently, but it's one that has intrigued me since I was a graduate student. So it turns out that, and neurons are a classic example of this, but they're not the only example, that as our bodies develop during embryogenesis, during our developmental process, certain cell types make the decision to what terminally differentiate. And that's important if you're a neuron, because you're going to have to develop a very, very complex cellular structure. You need these very elaborate dendritic branches. You need a very long axon that can project hundreds of times the size of your cell body. So it's a major engineering feat to put that together. The last thing you need, and this is retrospective logic, but it would seem like the last thing you would need at that point is the structurally disruptive process of having to divide into two cells. So what do you do with all that structure? Okay, you can butt off a little cell and call that cell division, but then you don't have the structure anymore. So all you've got is a little P. So as part of that differentiation process, neurons, and as I say, other cells go through this, become incapable of further cell division. As I often say, you get all of the neurons you're ever going to have by the time you're 1 year old. And after that, all you can do is lose them. That's a huge biological problem for our brains, because it means that if you do lose a cell, it cannot be replaced. But the logic behind my cell cycle work was that may be true, but cells never forget their evolutionary origin and will respond to stress very often by trying to divide to get out of the situation. And neurons are, in my opinion, no different. So during the stress of Alzheimer's disease, cells will initiate a cell cycle process. They will begin to synthesize the proteins needed to go through cell division. They will actually start to duplicate their DNA as if they were going to divide, but they are permanently blocked and they cannot proceed to the stage where they are able to go through a cell division process and create two cells from one. The original argument I made was that instead of dividing, they initiate a cell death process. And my hypothesis was that underlay the neurodegenerative process in Alzheimer's disease. That may be true, but recently a wrinkle on that idea has come forward, and that is that those neurons don't actually die. And in a complicated bit of biology, those neurons activate a process known as senescence. And that senescence process is a property of our cells that we believe functions in part to prevent cancer. Because when senescence kicks in, one of its features is that cell division is completely blocked. So all of that still fits with the data. The cells don't die, they simply enter the senescence process. And we do have evidence that that's true. But then in a really bizarre twist, my current hypothesis is that the senescent cells in the end don't die. But because of their senescence, they secrete factors
to trigger the death of other cells.
Trigger the death of other cells.
Oh, man.
And I'm going to say that as far back as 2007, I wrote a review in which I at least laid out that this was a possibility. And our more recent data suggests that this is actually what's going on.
So those senescent cells are producing the so-called senescence-associated secretory phenotype.
Correct.
This hodgepodge of different inflammatory chemicals that then take their toll on nearby cells.
Correct.
And drive the loss of those cells in what can become a vicious cycle.
Correct.
Nasty. So moving on to a different cell type, there are lots of types of glial cells. One of these is the oligodendrocyte. You've been very interested in these cells for some time. They are instrumental to myelination, which is basically wrapping myelin, this fatty sheath, around cells in order to speed the transmission of electrochemical signals, speeding the rate at which the brain can pass information on to nearby structures. And I think you were way ahead of the curve in pushing the importance of these particular cells. Can you tell us a little bit more about them and why you think they are so important to Alzheimer's?
Okay, so before I do, let me give a shout out to the person I think was the point of the spear in pushing the oligodendrocyte or myelination contributions to Alzheimer's. That would be George Bartzakis. He worked with imaging rather than with the cell biology itself, but was just passionate that one of the most reliable symptoms of a dementing illness is the loss of myelin in the tracts that service that area. Having said that, yeah, I have been fascinated by both the myelin and the oligodendrocytes, the cells that create it. And when I say both, I say that because the myelin, just as a physical structure, is extremely helpful to larger neurons. And the reason is that by insulating the neurons, they make the transmission of electrical signals much more efficient energetically. So the neuron spends a lot of its time pumping ions to keep itself in a state where it can be activated electrically. And those pumps cost chemical energy. The currency of energy is ATP. So they cost a lot of ATP. By coating the axon with a sort of an oily substance that won't allow ions to flow, it lets all those pumps take a break and it saves a lot of ATP. That's a structural help, but there's also a chemical help. So these oligodendrocytes also create nutrients that the axons use, and the axons in turn provide stuff to the oligodendrocytes. So there's a real strong cell-cell interaction that, at least in my way of thinking, the failure of those interactions contributes in major ways to the symptoms we recognize as Alzheimer's disease. And here again, like tau, we're not talking about something that is unique to Alzheimer's disease. So we've looked in other neurodegenerative diseases, and myelin abnormalities are involved there as well.
I suppose the most obvious of those is MS.
Yes.
Yeah.
But also ones that are less obvious, like this rare ataxia telangiectasia that I work on. It's a mutation in a part of the DNA damage repair pathway. Those people and those mice suffer from a loss of myelin and from oligodendrocytes.
How did you come to that form of ataxia? Was it through the oligodendrocyte?
No, that was backdoor. Once upon a time, I was actually a developmental biologist. I worked on pattern formation, and my focus was on the cerebellum. And it turns out that this particular DNA repair protein, known as ATM, when it's missing, leads to a really profound loss of cerebellar mass. So I was interested in it from its role in cerebellar development and maintenance. But then when I made the shift to Alzheimer's disease, I maintained an interest in DNA damage. And one of the reasons I maintained it was because the accumulation of unrepaired DNA damage does seem to be a driver of the aging process. So this protein ATM that I started working with because it kills the cerebellum has continued to interest me because it helps keep the DNA of our neurons intact.
We will come back to DNA damage and genomic instability. I'm sure of that. I wonder if you saw an analysis that came out a few days ago by Tony Whisker, who's at Stanford. Are you familiar with his work? He's doing all of this proteomic work looking at organ aging clocks at the moment.
Yep.
And he just did an analysis of the UK Biobank, which was very interesting. And basically, their suggestion was that people who have unusually aged brains, which I think they defined as at least 1.5 standard deviations above the average brain biological age of people of the same sex and chronological age, have a substantially greater risk of Alzheimer's. It's comparable to carrying one copy of APOE4. And they did this RNA sequencing analysis to look at the likely origins of the different proteins that made up their assessment of brain biological age. And about half of those were from oligodendrocytes, which I thought was further support for a lot of your focus. I thought you would have found that satisfying.
I'm very satisfied by it. Yeah, but look, let's be fair. Correlation does not prove causality. I argue that over and over again in my book. And live by the sword, die by the sword. I'm going to have to stick with that here. it's strong evidence that oligodendrocytes are tied up in the core biology of aging. We could posit that they help drive the brain aging phenotypes, or we could posit that they're a symptom. But either way, I think it's interesting and certainly merits further study.
Yeah, I think many roads lead to oligodendrocytes. But one of the interesting things about that analysis is that their estimates were based on adjustment for APOE genotype. And I know that you've done some work in recent years looking at how that genotype relates to oligodendrocytes and their function and myelination. What have you
Right.
found?
Well, it's a complicated molecular dance. So oligodendrocytes create myelin by extending their membrane and wrapping it around a small number of adjacent axons. That is a very highly specialized membrane that they use to make the myelin. We don't know exactly why, possibly because of its curvature. The membrane is very rich in cholesterol, and getting rid of cholesterol is almost incompatible with forming normal myelin. The problem that oligodendrocytes have is that they need so much cholesterol that they have trouble making it all themselves. And that trouble gets even worse as they age. So comes to the rescue from another brain cell type, another non-neuronal cell type known as the astrocyte. And these astrocytes make cholesterol and ship it off to the oligodendrocytes. Great. The problem that the astrocytes face is that cholesterol is completely insoluble, so shipping it off anywhere requires a transport. And the transport that they use is this lipid carrier protein known as apolipoprotein E. And that all has relevance because the APOE gene comes in three isoforms that differ in one or two amino acids, but those changes have a huge difference. So we know those isoforms by numbers. So one is called APOE2, one is called APOE3, and one is called APOE4. So APOE3 is the most common, and it carries cholesterol just fine, thank you very much. And astrocytes are happy, and oligodendrocytes are happy. APOE2 is protective, but APOE4 is a problem because it does not bind cholesterol as well, and so it is defective in that transport process. And our hypothesis is that loss of a supply of cholesterol is part of what underlies the biology of the loss of myelin in Alzheimer's disease.
That also maps to what we see when looking at global brain structure. So APOE4 carriers later in life have less white matter in the brain, and it's the
Correct.
myelin that makes it white.
Correct.
I don't think people will have fallen asleep when mentioning APOE, just because I think a lot of people already recognize that as being the largest genetic contributor to Alzheimer's risk in the general population. And to put some numbers to that, if you carry one copy of the E4 allele, then your risk is roughly threefold greater than if you were a 3-3 genotype. And if you carry two copies of the 4, so if you're APOE44, then it's probably something like a tenfold increase relative to 3-3. Is that right?
Tenfold or even more.
Yeah. So it's really significant. And tangent, but it's thought that the E4 allele was actually the wild type. And in certain conditions characterized by higher pathogen loads and so on, it might in some ways be adaptive, recognizing that our distant ancestors simply didn't live as long as we do. It's not like there's been selection against that, because by the time ApoE4's negative effects kick in, most of us have stopped reproducing. So
Correct.
it's no big deal. ApoE has roles in lipid transport, and that brings up metabolism in general. Another theme that has come up in your research is that of insulin resistance, which a lot of people speak about as being another contributor to Alzheimer's. And I imagine it's quite difficult to look at insulin resistance within the brain specifically in free living people. Is that a fair assumption?
That's a fair assumption, yeah. Yes.
But the current thinking is that brain insulin resistance as well as peripheral insulin resistance are both contributors to Alzheimer's.
Right. So our view on this, and I shout out to my former postdoc, now professor at Chinese University of Hong Kong, Kim Chow, who really spearheaded this work. What Kim found was that the elevation of blood glucose leads to a response, not just in the periphery, but in the brain, to make the cells more and more insulin resistant. And through a fairly complex metabolic pathway, that leads in turn to, and we'll get back, this is where the senescence argument comes from, that leads in turn for the cells to send out internal distress signals, which activates the senescence program and leads those cells to senesce, and as we said before, to perhaps inadvertently signal to their neighbors, it's time for you to ask for the check and check out.
Insulin resistance is something that is readily amenable to change in most people through lifestyle modification.
Correct.
We need to bear that in mind, and we'll come back to that later. But another contributor to insulin resistance is mitochondrial dysfunction. And my impression is that some people have become hyper-focused on mitochondria. I read Chris Palmer's book, Brain Energy, and at times I thought that he missed the forest for the mitochondria, let me put it that way. Clearly they do matter. What do you think their contribution to Alzheimer's is, and how big a player do you think they are? I'm sure that it varies between individuals.
I mean, I haven't read his book, but I would tend to be more inclined to promote the role of mitochondria in particular. But as you started, and I would totally agree, metabolism in general is a major contributor, and insulin resistance in neurons is part of that, but only part of it. I mean, I point out in the book, and I would really underscore here, our brains do not exist by themselves. I mean, our brains live in a body, and the brain doesn't work real well if the rest of the body is gone. And the reverse is equally true. Our bodies don't work real well if our brains are gone. But that's just a simple way of saying, look, we have to think about dementing illnesses, not just from brain biology, but from organismal biology, and it all matters. And I make that point in the book, but I would underscore it here while we're talking about metabolism, because the metabolic features of the brain are going to be driven in part by their own internal cell biologies, but they're also going to be driven by levels of insulin, levels of hormones, levels of other metabolites that are extrinsic to the brain. They come from the periphery. And all of that's going to affect brain function. And frankly, all of it is going to be part of the aging process.
So you think mitochondria are key players?
So getting back to mitochondria, yeah, I mean, I think that they're the energy powerhouses of our cells. They probably don't cause Alzheimer's disease, but once again, I think as they become less and less able to function at peak efficiency, the risk of Alzheimer's increases. I would accept an argument that it is, in fact, this slow degradation of mitochondrial function that is part and parcel of the aging process itself, and therefore part and parcel of one of the major risks, or the major risk, associated with Alzheimer's disease.
You spoke there about how important organismal health is to brain health. An example of that is that the brain needs an appropriate blood supply, which brings up the cardiovascular system. And within that, we can look at the density of the vasculature, we can look at vascular function, we can look at the effectiveness of the blood-brain barrier, and so on. Can you speak a bit about the importance of vascular health to brain health?
Oh, it's tremendously important. And only now, I think, coming into its own as a topic of study. Vascular dementia um, is very au courant these days. Um,
Why is that?
well, because it just, I mean, we're finally listening to our own data. I even pointed out in the book that some enormous a majority of cases of clinically diagnosed and pathologically diagnosed Alzheimer's disease have
Also vascular.
of vascular abnormalities. So it's very tightly integrated with Alzheimer's disease, and we have the clinical data, which is that exercise is a important and robust intervention that can reduce Alzheimer risk. And to get into a more pharmacological model, aggressive control of blood pressure, keeping pressure below 120, has profound effects on Alzheimer risk. So we're late to the table, but I think we're playing catch up about the importance of vascular factors. And I would argue it's probably not just hypoxia and ischemia that is a failure of nutrients, but I think that it is the accompanying hormonal health that comes from the other organs of the body that the brain needs to maintain its peak function.
It's a supply issue too,
Mm.
based on the analyses that have been done looking at the so-called population attributable risk factors. So the proportion of variance in dementia risk that's explained by different components of lifestyle and general health. High blood pressure is certainly one of the strongest risk factors that contribute